Okay, good morning, welcome. This is the first session of these talks in Introduction to Bioinformatics. And we start very high. We start with Professor Roderic Guigó. Roderic Guigó is full professor at the Universitat Pompeu Fabra. He's also the coordinator of the bioinformatics program at the CRG, the Center for Genomic Regulation. And probably all of you have heard the story of how the first draft of the human genome was obtained, and the race between the public and the private company to obtain this person here, Roderic, was there. In fact, he is one of the authors of one of the two publications that emerged from the first draft of the human genome. Okay, this is one example. You might have heard also about the ENCODE project, okay? A public consortium to identify all functional elements in the human genome. Roderic Guigó and his team has been involved in this project as well, a very huge consortium. And this is just two examples of relevant contributions. We could spend the whole lecture, I think, mentioning the curriculum of Roderic Guigó. So yeah, I will just say thank you to him. So yeah, it's a privilege that we can have him today with us, telling us the story of the history of bioinformatics. Okay, so thank you very much, Roderic.
Thanks a lot. You can hear me, right? Thanks a lot for your words and your introduction. Well, yeah, I will reflect a little what bioinformatics is, since it is a degree that you have chosen to devote the next 4 years, I believe, of your life. And I hope that you really enjoy these 4 years and you really believe that this was really the right election. I like to reflect on the relationship between biology and computation, and I will do it from a historical perspective. It's actually very difficult so it's actually very easy, because bioinformatics is a quite recent discipline. And because bioinformatics, as the name indicates, is the application of computational methods to biological problems, the discipline could not exist before computers existed. So we could trace, if you want, the beginning of this history when the first digital computers were invented. This was in the mid-40s. This is the ENIAC. It's the electronic numerical integrator and calculator. This is considered the first computer which it's on the path of the computers that you are using now. So it's a digital computer that can be programmed in memory. And this is a picture. This was a huge computer. I forgot. I think it's 9 meters long, like 2.5 meters high, and 1 meter wide. And these women that you see here were called programmers because they were programming the computer. And the way in which you program the computer at that time is by physically connecting the pieces of the computer. So in general, only the engineers that built the computer were able to use them, because to make a particular computation, in most cases ballistic trajectories in this time, you need to know which pieces of the computer to connect. And I think it's quite interesting that this happens more or less at the same time. I think this is a little bit later, that in a completely independent field of science, which is biology, Watson and Crick discovered the structure of the DNA, and essentially, right, realizing that the DNA was something that's very important. It's a polymeric molecule made of the repetition many times of 4 basic units, right? That I guess that you probably already know. These are these four nucleotides: adenine, thymine, cytosine, and guanine, that we represent by the letters A, C, G, T, right? And this is one of the important discoveries in the history of biology. This is the paper in which this was described. It's just 1 page in the Nature journal. So this is not. I guess that many of you already knew about Watson and Crick and the structure of the DNA, but I think it's also remarkable that this at this very same time, the very same year, I think, or more or less at the same time, this person, Frederick Sanger, actually obtained a sequence for the first time of the sequence of amino acids for the first time of a protein. Either this is the paper in which he describes the amino acids, right? The amino acids that form a particular protein, and I think it was the insulin from the cows. So this was very important because we had essentially two different codes, right? We had the code of the DNA, ACGT, and the code of the proteins, which is a code on 20 different letters because there are 20 different amino acids. And researchers at the time immediately realized that what would happen is that some way the sequence of nucleotides in the DNA influence or determine or gave rise or specify the sequence of amino acids in the proteins. And these were probably the best years in the history of biology or in the history of molecular biology, because the way in which this relationship was discovered was the results of many hypotheses, trial and error. So, because the most parsimonious hypothesis is that you say, well, the sequence of the DNA determines the sequence of the amino acids of the proteins. The problem is that the DNA is in the nucleus, and the synthesis of the proteins is in the cytosol, and the DNA never leaves the nucleus. Therefore, how come something that's in one place of the cell can determine the sequence of something which is in another place, and these things never interact with each other, and this led to the discovery of the messenger RNA. Right? The messenger RNA is the molecule that carries the information that's called messenger, right? Because it carries the information from the nucleus to the cytosol, where then this is translated to protein. And this was what at the end, right? At the end was known as the central dogma of molecular biology, but essentially is how we believe the information is transferred from the DNA to the proteins, which are the effectors of the molecular function on the cells and the structural components of the cells. And this was the work of many scientists. As I said, these 10 years that go from the discovery from the early 50s to the mid-60s were really extraordinary, and how we establish the molecular basis of life, and in particular the universality of this molecular basis. And this, in a sense, this culminated with the discovery of the genetic code. So the rules by means of which the sequence in the DNA, the sequence of nucleotides in the DNA, determines the sequence of amino acids in the protein. As you know, every 3 nucleotides in the nucleic acid sequence specifies unequivocally one amino acid in the protein sequence. And we have some bias about what we think a computation is. And when we talk about computation, you think about computers, right? And you think all computations take place in computers. But computation is a more general term. This is a computation, right? This process is a computation almost in a paradigmatic sense. Computation, right, is a process that takes an input and produces an output according to a particular set of rules in the computer, according to a particular program. Right? You type the name of an URL, if you ever type anymore, and then you get, as a result, a particular screen in your computer. So this is a computation, right? We have an input, which is the sequence in a particular alphabet, ACGT. We have a set of rules that convert this sequence into something else, which in this case is a sequence in a different alphabet. So this is a computation, right? It's another type of computation, but it's a computation. I don't think that I will have time today, and I don't know whether you will ever touch this during these 4 years, but there is an area of computation which is called DNA computation, in which DNA is used to solve computational problems, right? So this is not only a metaphor, it's actually, in principle, it's possible to build computers based on DNA that solve computational problems, right? Solve problems like optimization problems of how to travel between cities in the most efficient way. So this happens actually, all these things happen in the area of biology, but of course computer science was not inert either. There were things that were happening in computer science. Remember, as I said at the beginning, ENIAC, only the engineers that built the computer were able to program the computer because they knew how to connect the elements that need to be connected to compute anything that they wanted to compute. But by the mid of the 50s, something was invented, but it's called programming languages. This is one of the earliest and most successful programming languages in history, which is Fortran, which is still used in areas like physics. I guess that you don't study Fortran. But essentially, what happens with these high-level programming languages is that they separate the computer, how this is made, the hardware, right? What's called the hardware, from the usage of the computer, the software. So this was extremely important because then people that did not know how the computer was built and that had no clue on how the elements were connected, and the central memory, the accessory memory, the processing unit, whatever, right? They don't have any idea how these things are connected. They were able to write programs to make particular operations, right? And well, I don't know. I mean, at this point, right? At this point, there was no relationship between computers and biology because computers were used only in very specific relationships. But many of the terminology in molecular biology, right, borrows from the computer science because people already saw the parallelisms between another. And we talk about genetic program, genetic code, which are computational terms. We talk about transcription. We talk about translation. All these terms, right, are terms that are more related to computation on sequences of symbols than to biology. But now they became already part of the basic vocabulary of biology. So as I said, the computers, at that time, until the computers were very large, very expensive, and they have to build, it was complicated to build them. But then, of course, there were many advantages in engineering, in particular, all the components of the computers could be made smaller because transistors and then integrated circuits were invented. And the thing is that the computers become smaller, become cheaper because you less material to build them, and they become faster because the information needs to travel less distance, right, from one place to another. And therefore, this made that during the beginning of the 60s, the computers were mostly until that time were used in military research or related to military research, were actually introduced in banks, financial systems, and also in research centers. So think about this, only after half of the mid of the 60s, the computers were never used for research. And one of the first persons that used computers in biological research was this person. This person is Margaret Dayhoff. And we could say that in one way or another, this person is the father or the mother, actually, in this case of bioinformatics. If you have to choose a name in which really computers were first used to solve biological problems, we could actually relate to the work of Margaret Dayhoff. This is a computer and this is a phone. I don't know if you recognize this, but these were in this year. What did Margaret Dayhoff do? She did something very simple, right? She just collect, as I told you that in the 53, Sanger, by the way, Sanger is one of the few scientists that have gotten the Nobel Prize twice, once for method to sequence proteins, and then, as we will see, for methods to sequence DNA. So using the method of Sanger, many labs in the world have sequenced other proteins, right? Other proteins. And there were maybe by the mid-60s, maybe 200 or 300 protein sequences. But what Margaret Dayhoff did was something very simple, right? She just put all the sequences in a type book. There were no computers at that time, right? So she put this in a book. Essentially, each page of a book was the sequence of a protein, right? This is the sequence of a protein. Each letter corresponds to one of the 20 amino acids. But what happens when we put many things together, right? Different examples. You have an example of one thing here, one example of another thing there, and you are never able to put them together, so that you never see whether there is something in common or general between the examples of these things. So by putting the things together, by compiling a database, if you want a very early database, what Margaret Dijkhoff could do and her collaborators were to compare the sequences, right? Were to compare the sequences that were obtained in many different laboratories, right? The sequences were printed in the papers. There was no way of electronic communication, right? Just want to think this. They went to the libraries, they look at the journals. Well, this journal has the sequence. They type it in a typewriter, so the sequence of this protein. But they could actually compare the sequence, and what they found is that sequences that had similar function have similar sequence, right? Something that now we think is obvious. It probably was not obvious at the time. For instance, this is the cytochrome. This is a protein called cytochrome. Cytochrome, I don't know which one. Maybe cytochrome C, which is present essentially in all living organisms involved in the respiratory process. And this is what is called a sequence alignment. A sequence alignment, it's a matrix. It's a matrix representation of many sequences. Each row in this matrix is one protein sequence, right? It starts here, continues here, and continues here. And in this particular case, we have the cytochrome C, as I said, in different species, from humans to Neurospora crassa, which is a fungi. And what are the columns? The columns, and you will learn this during your bachelor, are positions, right, that in principle play similar functions or the same function in these different proteins, or in this same protein in different organisms, right? Different organisms. So the hypothesis underlying this sequence alignment is that there was an ancestor protein in an organism that was the ancestor of all these 4 organisms here, right? That had a sequence, right? A sequence through the evolution of which, right, and the speciation, etc., the sequences in the sequences in all different species have been generated. And just by the observation of this, right, this is already quite important from the biological standpoint, because you can see that the stars here, right, means that these positions have been conserved, right? We're talking about 600 million years, probably the distance that separates us from fungi, 600 million years. So in 600 million years of evolution, these positions haven't changed at all. I made a mistake again, which I don't know which mistake I made. That's something bad. Okay, it's here. So these positions really hasn't changed, and other positions instead really have changed or have even disappeared in some species. This already tells us something. This tells us that if I have to decide which positions are more important for the conserved function of this protein, this region here is probably likely to be very important for the function of this protein, because it hasn't changed in 600 million years of evolution. If it was possible to change this sequence without any impact in the biology, we would have seen variation, but we have not seen. It means that if we mutate one of these amino acids, probably this protein does not exert the function as well as it should. And anyway, I can go a little bit fast here, just because you will see all this during your studies. So Margaret Dijkhoff built these substitution matrices because she also realized that in these columns, sometimes some combinations of amino acids were more likely to be seen than some others, right? So it looked like during evolution, some substitutions of amino acids once for another were more likely to occur than substitutions from some other amino acids to some others. And she built this thing called the substitution matrices, which this scores simply means how difficult it is to observe that this has occurred during evolution. And for instance, -6 here means that substituting a tryptophan by a valine is very unlikely. But for instance, substituting glycine by a histidine, it's actually quite likely, right? So anyway, this allows for one of the early development, which I would say is the the birth of bioinformatics, there is, as the number of sequences was growing and was growing and was growing, the problem of building these alignments by hand, as Bijhoff had done, was becoming really, really complex. And then methods were invented for building alignments of sequence of automatic manner, taking into account the information of the substitution matrices. And I feel really honored to, this is Temple's. So this was on the 70s, right, when there was essentially very few sequences. And this was modified then with this Smith and Waterman. I am looking here at Smith and Waterman because I did my first postdoc when I finished my PhD with Temple Smith, which is one of the important names in the field of bioinformatics. So while we had already in the 70s many amino acid sequences, the sequencing of DNA was really complex and they remained quite elusive. It was not until mid-70s that methods to sequence DNA were invented. So by Sanger again in Cambridge and by Maxam and Gilbert in Harvard. And again, this is, as I said, this biology was going one way, computer science was going another way. They were already touching, but something that I think is remarkable, this happens essentially at the same time, more or less at the same time that the internet was invented, right? Internet was born at the middle of the 70s. So these are two things that had nothing to do, right? That had nothing to do, but they happened more or less at the same time. And something not as important as this also happened at this time is that I am actually, as you are now, I was starting my bachelor's. I studied biology. Yes, I started biology more or less at the same time that internet and the sequencing were invented, completely different. Why I am bringing this thing? Why is this relevant that I started this at the year? Well, because when I started studying biology, there was no sequencing and there was no internet. So what I am doing now, what I end up doing in my life as a scientist, I do programs to analyze genomic sequences that are used on the internet. What I mean is that it's not that when I started biology, I didn't know what to do. I didn't know whether I wanted to do zoology or botany or biochemistry or genetics or ecology. It's not that I did not know what I wanted to do. It's that I could not imagine what I would end up doing because what I am doing did not exist and nobody ever envisioned that could exist. Right? I'm not sure if I make myself clear because I think this is an important message that you need to understand. Maybe what you end up doing you cannot imagine now. It's not that there are things that you can imagine. Oh, well, maybe I will be an astrobiologist. I will do terraforming in Mars. Means converting Mars in a place where people can live. Right? Well, that may be science fiction. But you can imagine doing this. What I meant is that I could not imagine doing this because these things did not exist. Anyway, soon after, the first genome sequence of this virus, 5X174, was sequenced in the lab of Sanger. And by the way, this and then the amount of genomic sequence grew very fast afterwards. And by the early 80s, the first databases to store the sequence that had been generated were created. The first digital electronic databases, not the books of Margaret Bijhoff, right? And they were essentially the first one, and I want to emphasize this because I think that now, this is very popular now because you maybe some of you has seen the Oppenheimer. Have you seen the Oppenheimer, the movie? Seen that this happens in Los Alamos National Laboratory, right? So the database, the government of the United States decided to create the database to store all the DNA sequence in Los Alamos National Laboratory. Los Alamos National Laboratory, for those that have not seen the movie Oppenheimer, is where the atomic bomb was developed during the Second World War, the Manhattan Project. Okay? And why do you think that this database was set up in a place that was devoted to the research on weapons, on nuclear weapons? Well, I think it's because of the following. When the atomic bombs were dropped in Hiroshima and Nagasaki, well, it was very clear that one of the main effects of these bombs was not only the people that died because of the enormous temperatures that were created by the explosion. Also, many people were affected and died afterwards because of the radiation that remained. And how is the radiation affecting our bodies? It's through mutations, it's through provoking mutations in the DNA sequence. Therefore, when the database is to store all the DNA sequences were established, they were established in Los Alamos National Laboratory. Yes, actually, I'm talking a little bit about myself, but I can't. I actually work in Los Alamos National Laboratory. It's an amazing place. This is the laboratory, and I find very lucky myself of being able to have worked there for a few years, because this is an amazing place at the beginning of the genome project. One thing that I didn't mention at the same time that these databases were established, the first personal computers were also into the market. So you have to think that before 80s, for instance, when I started my PhD thesis in 81, but I did not have my computer, right? But we were terminals that were connected to a mainframe computer. There was a computer at the University of Barcelona. There was only one computer, and we have terminals through which we connected to this computer. Actually, we didn't have these terminals in our office. There were a room where there were 3 or 4 terminals for the entire university to go there. So it wasn't until the mid-80s that people start having computers on their offices. The personal computers were invented both by IBM and by Apple, right? So the computers that people could use in their offices and in their houses, although of course they were of limited value for most people until they became connected to the internet, right? Anyway, so what I wanted to say is that because the number of also at the same time, because of the number of sequences was growing very, very, very fast, the methods of Smith-Waterman that were invented to align sequences were not fast enough. Although they were very efficient, they were not fast enough, and new methods were invented with programs like FASTA and BLAST. And this is part of the folk history of bioinformatics. What is said is that this researcher, Doolittle, using these new established electronic databases in his personal computing and doing something that has not been done before, was comparing the sequences of all amino acids with the sequences of all amino acids with these programs that have been developed, right? So found this relationship between oncogenes and growth factors that has been escaped the attention of all the researchers until then. So oncogenes, as you know, are genes that are involved in cancer. And then this similarity between an oncogene and a growth factor. So let me explain what this is. This is the oncogene, actually. This is the oncogene. Well, maybe it's the other way around, actually. This is the growth factor, and this is the oncogene, anyway, right? So, and this indicates how similar these two protein sequences are. So illuminating, so this really provides great information about the molecular mechanism through which cancer is produced, right? It's an abnormal growth of cells. Also, sorry, again, it's myself, because this is actually, because in Los Alamos, at GenBank, and this was the reason because I went to Los Alamos, GenBank was established there, and there were the first genomic sequences there. There were also the first methods that analyzed genomic sequences to identify patterns that were functional. And I did this with Jim Fickett. Fickett is this person here, was my second postdoctoral advisor. So these are people that are very important in the life of a scientist. Temple Smith, my first postdoctoral advisor. Jim Fickett, my second postdoctoral advisor in Los Alamos National Laboratory. So these people are very important in my scientific life. Jim Fickett visited last week, and we took this picture. So what Jim Fickett did was the following. He looked at the few genomic sequences that were in GenBank. You know that genomic sequences code for proteins. We have seen it early. But in eukaryotic organisms or in prokaryotic organisms, it's not the entire genome sequence that codes for proteins. For instance, in our case, only about 2% of our genome code for proteins. Only a small fragments of our genome code for proteins. Therefore, what Jim did when there was very, very, very few sequences is develop a program that, looking at the statistical composition of the DNA sequence, predicted in which regions of the genome there were potential coding regions. And this is one of the first programs that analyze genomic sequence with the intent of identifying functional domains in the genomic sequences. And at the same time, other researchers have discovered, for instance, something important in the structure of the genes, the process of splicing. These regions that code for proteins, so you could imagine, right? You have a genomic sequence, right? And you have a region that codes for a protein. This region that codes for a protein does not do that in a continuous way. It's just fragmented. It's this piece, this piece, this piece. Through the process of splicing, the messenger RNA that's translated into proteins is generated. And the process of splicing means eliminating some regions to produce the final product that will be translated to proteins. Then researchers, looking at the databases, realize that at the boundaries, right, between the places that remain in the messenger RNA, the exons, and the regions that are eliminated, the introns, there was a particular consensus sequence in which essentially this is the last position within the exon, this is the first position within the intron. You see that we have always, right, the nucleotide GT. And this was observed again by looking at the information stored into the database. So I want just to mention this because I think it's the importance of having the databases. And the first this was purely computational analysis. There is no biology, there is no experimental biology developing this. So the fact that we had this motif in the border, which is this, right? In the frontiers between, the boundaries between the exons that code for proteins and the introns that are eliminated, right? This was done purely computational. And it's an important biological information that is produced by computational analysis. So the message here is that already early in the 80s, it was obvious that some problems in biology, because of the very first nature of biology information, could only be solved by using computational methods. So then we get into the 90s, into the Human Genome Project. This was a project that, by the end of the 80s, the methods to sequence DNA were sophisticated and mature enough that people thought that maybe it would be possible to obtain the entire sequence of the human genome. Just for you to have an idea, the human genome has a length of 3,000 million nucleotides, right? 3 times 10 to the 9, 3,000 million nucleotides. So the idea was to start a project that was predicted to last 15 years, to obtain a reference sequence of the human genome. Of course, each of us have a different genome sequence, right? But our genome sequence is very similar. Between two of us, there is one difference every 1,000 in this sequence of 3,000 million nucleotides, 3,000 million ACGTs. So we have to create a reference genome of the human genome. And at that time, it was already, now it looks silly because now, of course, we cannot do anything without computers. But at that time, it was not so obvious that to do biology, you need computers. But when the human project started, this is the white paper that was written to plan the human genome. It was very clear that without computers and computational methods, the genome project would be impossible. And I don't want to abuse of the parallels between biology and computation, but this actually happened. So the human genome started at the same time that the World Wide Web was invented at the CERN. The CERN is the European Center for Nuclear Research, for Physics Research, Research Nuclear. And this was by Tim Berners-Lee. This was invented, and you know what the World Wide Web is. You cannot live without the web. You are connected all the time. Well, maybe now it's not the web anymore. I don't know. It was until now, but still underlying almost everything that you use on the internet is this protocol, the double, triple W. Okay, and so the Human Genome Project actually lasted less than 15 years. Usually things last more than you predict. Right? I say, well, this will take 15 years, and it takes 30. Actually, this lasted less, and partially because what has already been said, right? There was a company that at some point, Celera Genomics, that said that this was a public project financed mostly by two institutions of the United States, the National Institutes of Health, which are developed to medical research, and the Department of Energy, which is dedicated to energy, and also a little bit by the Wellcome Trust in the UK. But most of the finding of the human genome came from the United States. And then there was a scientist called Craig Venter, and we use public funds, right? These are public institutions, public funds. There was a scientist called Craig Venter that said that this was a waste of mine, and that he was going to sequence the genome himself. He created a company called Celera Genomics, and actually he started sequencing the genome. And there was then the reaction on the public consortium was that it was really a danger that something as important as the sequence of the human genome would be in the hands of a private company, that they accelerated the public project, so that at the end the two papers, as you already know, were published together, right? This is by the public consortium, and this is by the Celera Genomics, and these are probably remember that the Watson and Crick paper was used one page, right? So in each of these articles, the first page is just the number of authors, right? Because there was a collaboration of so many authors, right, to do this project, that if you look at this, the first page is just the names of the authors, and I think these are for sure the largest papers ever published into these two journals, which are the most important journals in science, right? I think they run 25 or 30 pages or more, I don't know. And this is when actually happens the explosion of bioinformatics, right? Bioinformatics as a discipline, what you are studying, as I said, it's very new, right? Because before, so I think 2001, what year are you born? Were you born in 2001? No? 2003, okay. So even the human genome, you were born after the human genome, okay? So this is, I mean, you see, right? So this is, for instance, this is the proportion of articles in, this is very old, because this thing doesn't work anymore. Maybe now it has, I don't know, maybe it has already matched this other curve. But this is the proportion of articles in Medline. Medline is the database of all articles in biology and medicine, that contain the word, the word cancer, right? And you can see it's 6, 7, 10% of the articles in. So this is the main probably research area in medicine, cancer. And before the 90s, the words bioinformatics or genomics didn't exist. By 2010, we're already approaching to some extent, right? The frequency with which the term cancer appears in Medline. And now, of course, there is even cancer bioinformatics or cancer genomics are disciplines by themselves. Anyway, the human genome project, as everything, was only a prologue, right? It was the culmination of a project, but it's also the start of something else. And I want to finish my class today with which are the 3 areas in which genomics has evolved after the publication of the human genome, right? And these are, so essentially the human genome that was published in 2001 was the genome of one genome, was a referent genome. And as I said, each of us has a different genome, and it's in the genome that are encoded are the differences between us. So the reason to sequence the human genome was to have a better way to fight diseases, was a way in which The idea was that instead now, how do we fight diseases? We detect the disease, and then we try to treat it. The idea was that with the human genome, we will be able to anticipate the disease because the disease would be written in my code, and I would take actions to prevent that the disease appears, right? It's a very different model. That was the idea of the human genome. Rather than detecting and treating, anticipating and preventing, right? So, but the thing is that, of course, with one human genome, you cannot do very much, right? So you need to sequence the genomes of many people and see how the differences in the genome correlate with phenotypic differences. So there is an area, right? An area of research in genomics that goes into the characterization of the genomic variation in the human species. Then we knew, right, and we know that the genome encodes the instructions that determine our traits, right? My height, the color of my eyes, the color of my skin, the tone of my voice, the predisposition to particular diseases, etc., etc. But the way in which these instructions are encoded was totally unknown in 2001 and is mostly unknown now. So there is a lot of efforts to understand the function of the genome. And then, of course, we are only one of the millions of species that live on Earth. And another area of research for genomics is actually in the characterization of the genomes of all the species on the Earth. And I will briefly touch on these three areas. I just wanted to say that this progress in these areas has been possible mostly or almost uniquely, or well, not uniquely, mostly because impressive advances in the technology to sequence nucleic acids. So the human genome, 3,000 million bases, took 12 years to sequence. It involved hundreds of researchers around the world, and it cost $3,000 million. $3,000 million. Now, right, we can sequence hundreds of genomes in a day, of human genomes in a day, for about 300-400 euros per genome. So this has changed totally our the capacity that we have to not only to sequence genomes but to understand how the genomes work. And this is there is something called, maybe, I don't know, are you familiar with this Moore's law? Are you familiar? No? Never heard about this? So the Moore's law is a law that someone called Moore formulated early in the 60s, which is quite amazing, because early in the 60s there were very few computers. And he predicted the following. He predicted that the capacity of the computers double every 2 years. Every 2 years. And what does it mean that the computer? Well, I mean, there was a strict, I think it was the number of transistors or something. Anyway, it means that the speed of the computers double, let's say the speed of the computers double every 2 years. And you more or less see this, because if you buy a computer now and you buy it in 2 years from now, maybe it costs you the same, but it goes much faster, it has a larger screen, it has double of the memory, right? So it's not that the computer, the price maybe is the same, maybe it's more or less constant, but the capacity of the computer, and this is quite stable. This is quite stable. This thing, well, this is up to 2004, but still now. Although some people claim that the Moore's law is reaching their asymptote because of the limits, the physics of the limit, because of the laws of physics, right? The laws of physics dictate the limits to how small the things can be and how fast the speed of information can travel. Cannot be faster than the speed of light. And so the enormous progress in our culture, in our world during the last 60 or 70 years, right, more or less, maybe in part is thanks to the Moore's law. So this capacity that we have to double every 2 years the capacity, our computing capacity. Well, there are very few, and if we reach this limit, there may have consequences for. There are many of these doomsday hypotheses of what will happen to the world when we reach when the Moore's law stops being satisfied, right? Anyway, that's not the point that I wanted to make. The point that I wanted to make is that I think this is the Moore's law, double every 12 months, double every 7 months. So I think that more or less this, I don't know where is the Moore's law, I think it's, anyway, so the genomic information grows faster than the Moore's law, and there are very few things in the world that grow faster than the Moore's law. Anyway, so very, very quickly, so after the human genome was sequenced, and thanks to technological progresses, there was a project called the 1000 Genome Project that actually wanted to sequence genomes from diverse ethnic origins to capture the ethnic diversity in the human population. When the technologies further progressed, there were this idea of actually sequencing genomes associated to particular conditions or diseases, and this is one of the most important, the International Cancer Genome Consortium, with different countries participated with different types of cancers, sequencing by the genomes of about 500 or 1,000 individuals affected by cancer, the genomes of the cancer cells, right? And also the genomes of the somatic cells to compare of each individual. And thanks to the sequencing of all these many genomes through the cancer projects and through the others, that what we know is essentially most of what we know between the relationship between the genome and the phenotype, right? Our traits is through these association studies in which we are able to, by looking at statistical associations between individuals affected by a particular condition compared with controls, we identify locations in the genome that are associated to particular phenotypes. For instance, right? I sequenced the genomes of 500 individuals with diabetes and 500 individuals without diabetes, and I found the position in which all the individuals with diabetes have an A and none of the individuals without diabetes have a C. I could conclude with quite a strong probability that this A is related to diabetes. Of course, this never happened, right? This is an example of essentially these strong associations never happened. The associations are very weak. What will happen is that 55% of the individuals that have diabetes have an A in this position, and only 45% of the ones that do not have diabetes have C in that position, or something like that. Okay? And now this has escaped, and there is a lot of opportunities for bioinformatics outside research. Now, I think in bioinformatics, you don't need to think that until now, more or less, I had been associated bioinformatics to research. But that's not the case anymore, right? Now, bioinformatics is beyond research. For instance, now most of the genomes sequenced from human beings are actually sequenced through the medical projects of the different countries. So many countries already, as part of your clinical history, are sequencing the genomes of their citizens. The most advanced country is the UK, which probably I think it has more than 1 million citizens now that have their genome sequenced, and the idea is that the genome sequence can help to inform decisions in terms of diagnosis and treatment. Of course, this sets issues because when the data is for research, it's publicly available. When the data is not for research, it's not publicly available. But the fact that we learn things about the association between genomes and phenotypes is because we're able to compare many, many, many genomes. So there are now associations, for instance, like the Global Alliance for Health, for Genomics and Health, which tries to develop methods, bioinformatic methods, in which you could still use as many genomes as possible without compromising the issues of confidentiality that are associated to the fact that the genomes are univocally and defining each of us. So, understanding genome function, as I said, we know that the genome sequence determines the biological traits of individuals, but we don't know how these instructions work, how they are understood by the cell, and how they actually, by interpreting the sequence of the genome, are able to create the organism or to produce the organisms that we see. And one of the first projects, actually right after the completion of the human genome, has also been mentioned, is the ENCODE, the Encyclopedia of DNA Elements, which had as a goal the identification of all elements in the human genome that confer functionality. So most of these elements are actually discovered through sequencing. So this is something that I want you to, I don't have a lot of time, is that these methods for sequencing are invented originally to sequence DNA, right? But they can also, and then characterize obtain the sequence of the genomes of the individuals or of the species. But this can also be used these methods can also be used to characterize the function of the genome. Through something called functional genomics. I will not go into details. I prefer to have some discussion afterwards. Well, there are many functional genomics projects all over the world. This is some of them, and one of the most important projects now going on is called the Human Cell Atlas. So these methods to characterize the function of the genome require the use of millions of cells. So all the methods that were used experimentally, so it was impossible to characterize the function of the genome at one individual cell. But now there are methods available that can characterize the function, the function of the genome at individual cells, and this through the Human Cell Atlas project. And let me skip this because I want to go to the end. The third area in which there will be a lot of activity during the next years, and maybe something that for you is of interest. So, as I said, the human genome the human is only one of the species in Earth. We don't know how many species on Earth there are. There are about 1.8 known million eukaryotic species. There is a project called the Earth Biogenome Project that aims to sequence all the genomes of all these about 1.8 million eukaryotic species during the next 10 years, right? So this is on the context of the threat to the biodiversity that we're witnessing, with many species being threatened, and this is a sort of network organization in which groups from all over the world work on on the sequencing of a specific species, those on their territories or particular taxonomic groups. Here we have in Catalonia, we have the Catalan Initiative for the Earth Biogenome Project, which has sequenced so far 40 genomes. The idea is to sequence all the genomes that live in the territories. And this is actually what we have, as I said, we have, so it's more than is in this figure, it's about 40. And why do we want, what is the importance of having the genome sequence of all the species on Earth? Well, it's to understand again how the genomes encode the traits. For instance, there are many animals that have the capacity for regeneration across all the metazoan phylogenetic tree. But there are mammals that can regenerate and mammals as us that cannot regenerate. Well, deers can regenerate com es diu, els corns, no, well, whatever, you understand, right, what I mean? So, if we have the genomes of all these species that are able to regenerate, we will be able to understand what are the genomic determinants of the capacity to regeneration, and then maybe, right, we can, now that we will have in the future the tools to edit the genome and to modify, maybe we will be able to, at some point in the future, engineer humans that will be able to regenerate as well. So let me stop here. I had other things that I wanted to mention, but I think that it's best if I stop here. Okay. Thank you.
Okay. Thank you very much, Roderic. Very nice talk. I hope it was inspiring to all of you. So, time for questions. We'll start first with persons that have priority for this session. So please raise your hand and I will come to you. Yeah.
What do you think is the biggest progress of bioinformatics so far?
What is the biggest problem or progress?
Progress.
Progress. Well, it's difficult to answer this question right now, because now, if you asked me this question a year ago, I could have answered a few things, right? For instance, I think, to me, within what I understand by informatics, the methods to align sequences, for me, were extremely important, because sequences carry a lot of information about biology, and these methods were actually what makes, these methods are unique to biology. But now, of course, we have this explosion of the artificial intelligence and the machine learning methods, right? These methods are not important only on biology, but are very important also on biology. You have heard also about ChatGPT, right? This you know, but you may not have heard. You may not have heard about AlphaFold. Have someone heard about AlphaFold? So one of the main problems, one of the typical problems, one of the early problems in biology, is to predict how the proteins fold from the sequence of amino acids, right? I already told you that proteins are a sequence of amino acids, but the DNA is more or less a linear sequence, although of course it has the chromatin and all this, but the proteins fold in different folds, and it's the folding, the way in which they fall in 3D, the shape that they take in 3D is what matters for their function, right? This is one of the early problems in bioinformatics, and there has been efforts. There is even a competition called CASP. We ran for 40 years, right? And there were methods that were better, worse, but it was impossible really to get good predictions. Now there is a method called AlphaFold, which is based on this, what is called these foundational models, right? These foundational models, and that outsmart any of the methods, actually do almost perfect predictions. So this is really amazing, right? A problem that we have been trying to solve for 40, 50 years, and then artificial intelligence is able to solve much better than we could, without clearly understanding, right, which are the mechanisms by means of which this program solve the problem. But the fact is that they solve the problem very well. I hope I have answered your question.
You have mentioned that when you were studying in university, the field of bioinformatics hadn't appeared at all. There was a specific person who motivated you to be involved in this field, or was it more because of the relationship between the topic you studied and bioinformatics?
Well, I mean, this is a good question, because things in life sometimes it's not always. I could tell you something and then tell you something different. I could tell you a sort of scientifically based answer, but I could also tell you a personal-based answer, and it's a mixture of the two things. I studied biology, but I didn't like to work on the lab. I was very bad. I did many mistakes. Also, I didn't like the fact that I had to, if you do experiments in the lab, you have to be very disciplined. You have to go from 7 to 8, and I thought, well, I mean, I like something that gives me more freedom, and I actually like it a lot. Things like logic, statistics, and all this when I was studying biology. So at the end, when I decided to do my PhD thesis, I did already my PhD thesis using computers, but not on bioinformatics. It was called in evolutionary ecology with a professor, Jordi Ocaña, that he had probably the first person at the University of Barcelona or in biology that had made a computational, himself already, a computational thesis where simulation models and things like that. So I finished my thesis, and I was going out with a woman that had finished the PhD much earlier than myself, and she had gone to the United States, to New York, to do a postdoc, right? And then, of course, I know we were like 1 year, at this time, 1.5 years apart. So just remember, there was no email, there was no Twitter, there was no phones. We wrote letters. I know what letters are, but something that you write by hand and you put in an envelope, stamp, you send it, and 10 days if you are lucky, or 2 weeks, or maybe a month later, the other person receives the letter, she writes back, and all this is great because I could do whatever I wanted. I didn't have a Twitter say, well, what are you doing tonight? No, because it was impossible. It's happiness. Anyway, the fact is that I was looking for a place to do a postdoc, and I didn't find anything in New York, but Boston is not too far from New York, and in Boston I saw Temple Smith working on this project, and I said, well, I like this thing of the, because he said, well, I'm interested in the syntactic and semantic relationships on the DNA, and I thought, well, it was a mixture of, I wanted to go close to where Monse was, and also wanted to do something related to my abilities. So sometimes to do the things because of life is just one thing.
Okay, so with all the sequencing that has been made, is there any evidence that suggests that the non-codificator zones of the DNA have an effect on the expression of genes?
Yeah, that's a very good question. So, I mean, there's a thing, long non-coding RNAs were essentially, you mean the non-coding region of the genome, right? So, yes, so as I said, the proteins, which are thought to be the main effectors of biological function, are encoded in about 2% of the human genome, right? The 98% of the human genome does not code for proteins, right? Does not code for proteins. It's these regions that are eliminated, the introns, or the regions between genes called intergenic. And actually, even these regions have sometimes called junk DNA, DNA that has no any function, right? This is not true. This is not true. Many of the regions are involved in the regulation of the expression of the genes. So as I said, we have these regions that code for proteins, 20,000 in our case. We have the same DNA in all cells, but our cells are different, right? The skin cells are different than the hair cells or the brain cells. Why is this if we have all the same genome? Well, it is because these genes do not work, all of them, in all cells. In the brain cells, some set of genes works. In the skin cells, a different set of genes works. And what does it mean that the gene works? It means that this DNA region is transcribed to messenger RNA, which is then translated to protein. The regulation of why some genes work in some cell types and not, why some genes are expressed in some cell types and some are not, is through regions that are non-coding. It's called the promoter regions upstream of the genes, or the enhancer regions away. Then there is another class of genes that were discovered and maybe, well, it was becoming evident that they were important like 10 or 12 years ago or 15 years ago, which is called long non-coding RNAs. These are regions of the genome that are transcribed to RNA, but they do not code for proteins, right? So there is a lot of discussion whether these things are functional or not. Some people believe that they are functional, some people believe that they are not. It is very clear that some of these long non-coding RNAs are very important. There is one very famous called EXIST. So you have two X chromosomes. We have only one X chromosomes. It's very important that the dose of the genes is maintained. It is not, you would not live well if you have twice as many products of the chromosome X as we have. So one of your chromosome X is mostly inactivated. So only one of the two chromosomes works, although you have two. And the inactivation of this one of these two copies is through a gene that doesn't code for protein, which is called EXIST. So there is evidence that some of these regions are functional.
Do you think artificial intelligence will make humans obsolete in bioinformatics research?
That's a good question, because this is something that I wanted to, and this is something that you need to. This is a very important question, because I think that your life in the future in research will be different. This is a very old paper, well, not very old, but it's 15. So we all think that this thing of the artificial intelligence and all this is something new that's emerging now. It is not true, right? It is very old. It's 40. But anyway, but now really, because two things: because there are a huge amount of data that we had connected through the internet. That's the important thing. And second, because the computers are very fast. This is the main difference, right? The main difference is that now we have the data available is incredibly much larger than before, because the computers are faster. But there is already work done many years ago that suggests that many tasks that scientists do, that researchers do, can be automated and done by robots. So this is a paper by Ross King. They developed a robot that did the following. There are many robots in the laboratories, right? There are many robots that do things, right? And you program them. No, this robot did the same. The robot did an experiment. And based on the results of the experiment, the robot itself planned the next experiment, right? So it was a very simple experiment. It's a growth, it's yeast growth, right? So the idea, of course, it's very interesting if you read this paper because, well, this robot works 24 hours a day, days and nights, 7 days a week. It does not complain. And it works as well as PhD students in terms of, because they make a comparison how well the experiments were done by the students and by the robot. This was many years ago. So what I'm going to, I think that you will need to interact with robots, not the inert robots that we are taking now, but robots that will have some degree of reasoning and of taking decisions by themselves, right? There are two things, right? One thing is you have ChatGPT, that's not a robot. You ask him or her, sorry. You ask the program a question and answers well or bad, but it doesn't take any action, right? ChatGPT doesn't do anything. This does, right? This does, because the robot thinks or whatever, and the result of this thinking takes an action. In this case, this is not very relevant because this is yeast growth. And at that time, there was not the capacity of DNA editing. But now we have the capacity of DNA editing. Now think, there is a robot that's autonomous, that can take decisions and can decide for ourselves whether editing the DNA, our DNA. So there are many, also I think that we're entering an area in which there will be many, many ethical problems that will be very, very interesting. But in theory, right, you could imagine that there are robots that can perform CRISPR, you know CRISPR, right? CRISPR experiments. And if they do it in cell lines and all, that's not very important. But if they do it in, I don't know, in embryos, human embryos, that's a different thing, right? And if they can make the decisions by themselves without human interaction, that's something, right, that we need to think whether this is a possibility of a science fiction. Yeah.
Okay. Thank you very much.
Thanks.
I think we have to leave it here. It's late already. I don't know if we should be worried now.
No, sorry, I don't want to worry you. But let's think a little bit. I mean, what I said is probably silly. This will not happen, right? But I think that these things like ChatGPT and all these things will probably are here to stay. And you need to not. I'm too old to learn these things, but that's what you have to do to learn them.
Okay, so let's thank again Roderic.